Hello, bio friends. I'm going to make you just a quick video to answer some of these questions about hypertonic, isotonic, and hypotonic solutions. First, we're going to start with energy, though, from the beginning of the chapter. Kinetic energy versus potential energy. Kinetic energy is the energy of motion. Potential energy is stored energy of an object that has the potential to be converted. So kinetic energy are things like light, heat, wind. Potential energy, a food item, a candy bar, ATP, a swimmer on a diving platform, a skateboarder at the top of a hill. So that is potential energy. True or false? Let me move it up here. Every energy conversion will decrease the entropy of a system. If false, make the statement true. So every energy conversion gives off heat, and that heat creates disorder, creates more entropy. It goes from, if you will, a gas in your car to the heat and the CO2 in the universe, from a battery to the heat that your game gives off. So it goes from ordered to less ordered, which is an increase in entropy. So that is false. It is possible for a candy bar to have potential energy for two different reasons at the same time. This is a tricky one. So yes, two different types of energy at the same time. It has energy in that if it's sitting on top of a shelf and it falls off, it has potential and kinetic energy because it is an object. Also, if you unwrap it and eat it, you're using the energy to create ATP, which helps fund your cellular processes. So it has two different kinds of energy. It can fall off as an object, or you can eat it and gain energy in the form of ATP. Okay, we are going to talk about calories. So if you look in your book, or if you look somewhere else, then I think I might have it. It is from your book. It is kilocalories and calories. Essentially, what we call calories are actually kilocalories. I can drag it over here real quick for you. So essentially we have a baked potato. Oh, I don't know why my computer says that. We have a baked potato here, and it has 220 what we call calories. Those are actually kilocalories. That is, we just say calories with a capital C, but food calories are actually kilocalories. So the answer to that one is going to be, so essentially what we have is 220 kilocalories for the potato and 220,000 calories with the lower C. So just to get those straight, sometimes you'll see kilocalories on things and you'll think, oh, that's not really a calorie. It is really a calorie. That's how we measure it. Complete the following about ATP. The energy contained in the food we eat directly powers all of our cellular activities. False. That is something that it's just the way we say it, but that's actually not what's happening. What's happening is the food that we eat generates ATP, and ATP directly powers our cellular activities. So you eat to get ATP. ATP is sort of the intermediary, if you will. It's the money, it's the currency that we use to fund the cellular activities. A cell pumps certain ions against their concentration gradient. So remember, we want to go from, if you're in a high concentration, right, you're shoved in the closet, you want to go to the lower concentration. This is a natural process. If I have to work to shove you in the closet, that takes work, that takes energy. So if a cell pumps ions against the gradient, shoves you in the closet, in order to maintain gradients, what type of cellular work would this be an example of? This is an example of transport work. Sometimes that takes energy, sometimes it doesn't. But it's transport. Any cell pumps that pump anything against a concentration gradient will basically take energy. If you're going against the gradient, it usually is going to take some energy. Phosphorylation, which is the process of adding that third phosphorus onto your ATP. If you remember that from class, that phosphorylation goes from AD to being diphosphorus to ATP. Again, that process looks like this. So there's your triphosphorus going to diphosphorus, and that gets recycled in your cell. Okay, moving on to enzymes. We talked about enzymes. Remember, it's basically the third girl in a fight. Essentially what that is doing. So a protein that speeds up a chemical reaction is an enzyme. The amount of energy needed to perform a chemical reaction is activation energy. That's this slide again. You've got the activation energy. This is how much energy it will take, how much heat, how much whatever to get from here to the products. But if you have an enzyme, it doesn't take very much energy at all. You are adding that enzyme to the situation, and that is speeding up the reaction, but it is not used in the process. Move this out of the way, maybe. Maybe not. Maybe I'm not going to move it out of the way. Okay. Really move it out of the way. There we go. Okay. The relationship between an enzyme's active site and a substrate looks most like a comb and hair, a car and driver, a scarf and a hand, a lock and a key. So it tends to change shape when that substrate goes in. So this is a lock and key mechanism. So an enzyme and a protein, an enzyme's active site and a substrate is often called a lock and key mechanism. Oh, here it is. I have it here. Illustrate the without an enzyme and with an enzyme. The figure below illustrates the difference between a reaction with and without an enzyme. Briefly explain your answer. Will it still occur without an enzyme? Yes, it will. It will be more unlikely to occur, and it will take a greater amount of heat or other kinds of energy to get that from reactant to product, but it would eventually happen. It will just be extremely slow. It will require a higher amount of energy activation to react. Okay, membrane function. This was what we did in lab today. There are 4 functions that membrane proteins can perform. This is the actual membrane, the phospholipid bilayer that we've talked about. Looks like this, the phospholipid bilayer. So you've got cell signaling, you've got cell-to-cell recognition, you've got transport, you've got enzymatic activity. So you can see all those. Here's your transport right here. You've got transport in and out, you've got cell signaling, you've got cell-to-cell recognition, you've got enzyme activity, you have just the way it is attached to the cytoskeleton. Match the following terms. So diffusion of water across a selectively permeable membrane is called osmosis. The movement across a cell membrane that requires no energy is called passive transport. Movement of a substance from high to low. There's no semipermeable membrane. That's diffusion. This is I spray something smelly, it diffuses. I put a drop of food coloring, it diffuses. There's no potato or there's no semipermeable membrane. That's the difference between diffusion and osmosis. One has a semipermeable membrane. When there is a difference in the amount of substance from one distance to another, that's called a concentration gradient. The high concentration is where there's a lot of material. The low concentration is where there's not a lot. So when there is a difference in the amount of substance across a distance, it's called a concentration gradient. The movement of a substance from a lower concentration to a higher concentration takes active transport. It takes me energy to shove you back into the closet against your concentration gradient. Okay, kind of a little review from the lab today. So isotonic, when 2 solutions have the same amount, net effect on a plant cell, nothing. Net effect on an animal cell, nothing. Everybody has the same concentration. Hypotonic is when a solution has less dissolved solute when compared to another solution. So this hypotonic solution would just be your water solution in the lab that we did today. So the effect on the plant cell is the cells would swell and become kind of turgid, meaning they are not wilted, they are thick. Effect on an animal cell will swell with water and burst. This is what happens if you give someone an IV of pure water. Please don't do that. The hypertonic is a solution that has more dissolved solute when compared to another solution. So this would be a salt solution with something in it or a sugar solution with something in it. The plant cell would lose water and shrivel up because water is trying to go to the hypertonic solution and the water is trying to dilute things. The effect on the animal cell, an animal cell would lose water and shrivel. If you look at this right here, the sugar molecules would diffuse throughout the tube until equilibrium is reduced. So basically, The water will just